This episode is produced with support from the National Centre for Biological Sciences.
Dear listeners of the Nature India podcast, welcome back to Biolore, this series where we are digging out some very interesting stories from the history of biological sciences in India. Digging is a good metaphor for this episode. You will soon know why I'm saying that. I'm your host, Shubhra Priyadarshini, and this is the Nature India podcast. We are still basking, by the way, in the aroma of basmati rice from the last episode. Today, we are going a little further down in history with gene detectives who basically leave no stone unturned, well, pun intended there, to look for signs of ancient life.
On the sands of time, stories unfold, treasures from the past to modern lore. Every leaf, every cell, life secrets they tell. Biolore.
Imagine holding in your hand a bone from an extinct animal, or a centuries old museum specimen, or a trophy from long ago hunts. What secrets do you think may lie buried within these relics of the past? What if I told you that these remnants contain something remarkable? A time capsule of genetic information that can reveal lost histories and shape the future of conservation and human well being. As genetic sequencing advances, researchers are now able to use ancient DNA to answer questions about evolution, extinction, and even disease outbreaks. What exactly is ancient DNA and where does it come from? It could actually come from very surprising sources: animal trophies, museum specimens, fossils, old collections, or buried deep in the permafrost. These samples give us a glimpse of the species that existed long before their populations declined or went extinct. Scientists are also using ancient microbial DNA from the bacteria Yersinia pestis to track the evolution of diseases like the plague, tracing them back to their origins and understanding how certain genes may have increased or reduced the risk of infection. A prime example is Africa, where researchers are now using historical DNA from black rhinos and lions to make critical decisions in conservation. This helps guide efforts to protect species that are currently endangered. But none of this would be possible without recent breakthroughs in genetic technology. While ancient DNA was first extracted very recently, just about 30 years ago, even more recently have we been able to study entire genomes thanks to the high throughput sequencing technology. This technology has transformed fields like anthropology, archaeology, and evolutionary biology. Of course, this field isn't without challenges. There are ethical concerns when extracting DNA from ancient remains. Whose remains are these? How do we respect the cultures and histories tied to them? Stay with me as we journey to the Skeleton Lake in the Himalaya, India's tiger lands, and to the Harappan ruins, and explore how the dead are speaking to us in ways we never imagined. Molecular biologist Uma Ramakrishnan is precariously perched on a ladder. She is in the Chhattisgarh home of a member of a former royal family. Mounted on the wall in front of her is a massive taxidermied tiger head. She gingerly balances a scalpel and a raft of chemicals and slowly starts prying apart a slice of the hardened tiger skin in her makeshift workspace.
And in fact, like one place, we didn't even have a good ladder. So we had to just take the ladder they had. And it was just a ladder with just rungs, thin rungs. So imagine, balancing on that, with a scalpel, with the alcohol, with the bleach, with, wipes and the tubes and not spilling anything. It's a little challenging.
Uma is a molecular biologist at the National Center for Biological Sciences, and for most part of this year, she has been moving around, peering at over 70 tiger trophies, like this one belonging to a former royal family. She takes DNA samples from these long dead animals. After they are sealed in vials and taken to a special lab, these ancient or historical DNA samples will tell us how the tigers once lived and prowled in these landscapes.
I use endangered species as models to understand extinction.
Mm-hmm.
What hits you first is, man, so many of these animals used to be there, and now there are so few of them. One part of conservation science is protecting what's there through ecology, protecting of habitats, creation of corridors, right?
Mm-hmm.
Another part of you wonders, there used to be so many that are now gone, yet these trophies remain. So can we learn something about the past from these trophies? Tigers are recovering in some places, and there is a very like a ambitious and a movement in conservation towards what we call restoration or rewilding. How can we help animals to colonize more areas, increase their populations, right?
Mm-hmm.
So for that, it would be great to know what was the genetic diversity or who were the animals in places where they are now extinct. And the only way to know that is to go back in time and to look at these trophies. Like, for example, in Chhattisgarh, there are no tigers today, but there are a lot of trophies. So we can use those trophies to understand, routes of movement. Did we lose some specific genetic variants when we lost the Chhattisgarh tigers? Or are they just simply the same as Madhya Pradesh tigers or neighboring state tigers? It's just totally fascinating and cool, right? There's something really interesting to learn, not just from a story perspective, but from a applied conservation perspective. We can
Mm.
use this information to plan conservation. And that's, for me, that's really important in all the work I do. These trophies, they provide a window into the recent past and the time period where humans have had a huge impact on biodiversity, last 200 years, right? 200 to 500 years.
We have decided that there's three target species: the a placental mammal, the mammoth, the marsupial mammal, the thylacine, and a dodo, which is a bird. And for each of these, there are different technical, ethical, and ecological challenges that we will need to be able to overcome to bring them back.
That's American evolutionary biologist Beth Shapiro. She's an ancient DNA adventurer who led a group that sequenced the dodo genome. She's the chief scientific officer of Colossal Biosciences, a biotech company gearing up to bring back the woolly mammoth, the dodo bird, and the Tasmanian tiger or thylacine. Beth saw the scientific innovations that made the field of ancient DNA possible.
These really incredible, capable scientists that are genuinely contributing to developing tools that will become part of that next generation toolkit for biodiversity conservation. Ancient DNA really has been driven by scientific innovation. In the very beginning of the field, we were using, it's now old school technology, but it was brand new when I started, where we were amplifying targeted bits of DNA using PCR, and we needed to amplify fragments that were longer than 100 base pairs at a time, 100 letters of the DNA code. And it turns out now that most DNA that survives is much shorter than that. The fragments are much too short to have been amplified. So there was a big stepwise innovation in science where we call it the next generation sequencing revolution, where you don't need primers, you can amplify all the DNA that's preserved in a sample. And this made it possible to sequence whole genomes, to get DNA from things that weren't preserved in ice, to do environmental DNA work. Really opened up a whole new universe of possibilities for working with ancient DNA. And the same has been true for computational innovations. How are we able to make sense of the hundreds of billions of short fragments of DNA that we can extract from a bit of dirt or a mammoth bone? How can we use all of the genome sequences that are available in the world today to identify the regions of the genome that make a mammoth woolly compared to a not woolly Asian elephant, which then we can target using gene editing technologies to take cells of Asian elephants and transform them into cells of something that looks more like a woolly mammoth.
All right, let's bring it back to present day India. Scientists are also diving into this cutting edge technology to unravel the complex ancestry of modern Indians. They are using ancient DNA to piece together the rich history that connects past and present.
So, ancient DNA is a field, it's a tool, it's a genetic tool that takes you in the past. It's like time machine.
That's Neeraj Rai, ancient DNA group head at Birbal Sahni Institute of Paleosciences in Lucknow.
For example, if I'm working on a sample of an individual who has lived about 5,000 years before present,
Mm-hmm.
so by their genetic sequencing data, we can really go into past. We can see the movement, migrations, and the history of that person about 5,000 years before present. So it's
Mm-hmm.
really like a time machine. South Asia is extremely diverse geographical region of the world, and every individual of this region has multi-layered ancestral components.
Right.
So just to de-layer the ancestry, and just to understand the exact source of ancestry that we have in the present day scenario, you have to have ancient DNA genetic data.
Right.
Without that, you can't really understand the past demographic changes and the prehistorical and historical migrations that took place in India to such a diverse region of the world.
Right.
So my group is mostly focusing to generate data from the ancient remains that is lying everywhere in the country.
Well, that sounds like quite a treasure trove. What is the reason that India doesn't have as much data as anybody else?
Because nobody had cared. And
Hmm.
working on ancient DNA is a really tough and challenging and a frustrating job without the expertise, because not much people are working in this field. For example, if you are sequencing 10,000 human genome from a region like South India or North India,
Mm-hmm.
you are not going to get any information,
Hmm.
because that information could provide you like hundred of samples. Maybe if you're sequencing 1 lakh samples, there is also
Mm-hmm.
not going to give you information about their population history. And at the same time, if you have 10 ancient genome data from North India, so you can really reconstruct the entire population history of North India that goes back to 5 6,000 years before present. So
Mm-hmm.
this is the power of ancient DNA. And considering the fact that India is such a diverse region, if you're sequencing 1 lakh, 2 lakh, maybe 10, no, no, 10 lakh samples, you're not going to get anything. Like you will just have a primary information about their diversity, but
Mm-hmm.
not about their ancestral source, how the ancestry was formed and how they have migrated.
Now, collecting samples is just one piece of the puzzle. The real challenge? It's the quality of the DNA. Ancient DNA is often fragile, degraded by time and the environment.
There's DNA damage, so you have to be very strict in how you do the analysis.
Let's now bring in another ancient DNA adventurer, geneticist Kumaraswamy Thangaraj at the Center for Cellular and Molecular Biology in Hyderabad. He explains that the tropical climate can be really tough on DNA. It tends to break DNA down into tiny pieces. And that's not all. The samples often get contaminated with bacteria and fungi, which makes things even more complicated for researchers trying to extract usable DNA. It's tricky, but scientists have developed clever techniques to work around this, pulling valuable information from even the tiniest, most damaged fragments.
In the tropical country like India, is not preserved properly, is exposed to all kind of environment. Therefore, there's a degradation of DNA, deamination, and also the contamination with bacteria. We have to use different kind of technique. There's no one particular standard protocol for isolating DNA from different biological, ancient biological materials. So one has to do every time develop a method for isolating DNA.
Now, while the challenges are real, they also create unique opportunities. For example, right now, Neeraj is working with samples from iconic sites like Mohenjo-daro, Harappa, Lothal, and Dholavira, also specimens from the Himalayas and southern India, and even Sri Lanka and Nepal. One of his most favorite ancient DNA story is about an Indus Valley woman buried in Rakhigadi village in Haryana.
about 5,000 years before present, most of the Indians were having that common ancestry. So that individual had only two ancestry. One is coming from this ancient Iranian hunter-gatherers that is quite common in all the present day Indians. And second ancestry was coming from Andamanis hunter-gatherer. We model that ancestry as ancient ancestral South Indian ancestry. And we did not find any Central Asian or European ancestry in that individual. So the date of that individual about 2200 BC, maybe the 4200 years before present. And in the present day Indians, every Indian were having that Harappan ancestry. Okay. And after the decline of Indus Valley civilization, there were a lot of demographic changes have taken place. For example, the populations from South India, they started mixing with the population to the IVC and descendant of IVC, in fact. And then another wave of migrations that have taken place from Central Asia and mostly from Central Asia and Iran. So they brought a lot of this steppe-related ancestry. So this is a major question in India. Like everybody wants to know, like when this mixing has taken place. So currently we have data, we have huge data that suggests that until 1500 BC or maybe 1400 BC, we are not finding any steppe-related ancestry in India or Indian subcontinent.
And all of these findings can also stir debates.
So sometimes these genetic findings also become controversial.
Right.
If a group of population is mixing to some other group, so they also brings a lot of ideas, lot of cultural materials with them. So we have not found so far any cultural artifacts or any cultural idea. Also this mixing is like quite gradual, so we are not finding that there was a migration, huge migrations, and that turned out to be a major genetic impact to the Indians. If you go back to 3500 years before present, so most of the North Indians, like population from Kashmir to the population to Madhya Pradesh, they were having same genetic ancestry, and that genetic ancestry was quite common. That we call this ancient Indus Valley ancestry. It is spread up to Kashmir Valley also. And then suddenly, like after 1500 BC, some kind of migration has taken place from Central Asia. They brought steppe related ancestry. So that ancestry is everywhere in India. But the question is that when that ancestry was brought to India, if that ancestry was brought to India before 1500 BC, then maybe the Indo-European languages might have brought by those people. This Rig Veda might have brought by those people. But we are not finding any genetic signal that is coming to India before 1002 or maybe 1200 BC. So Rig Veda, horses, and this Indo-European languages, all these three components then must be indigenous. So
Right.
yeah, question of debate.
And a closer look at DNA of the dead can often throw up surprises, like the unexpected discoveries in 2019 from the Rupkund Lake, eerily known as the Skeleton Lake or Mystery Lake, at over 5,000 meters above sea level in the Himalaya. These hundreds of skeletal remains found scattered all around the lake baffled researchers, including Thangaraj and Neeraj, for years to come. What made this site even more significant was that the Rupkund study marked the first ancient DNA data ever obtained from India.
But the question was that nothing was there. How did the people migrated there, and why such a huge number of skeleton was lying there? There was a lot of stories. People thought that there was traders who migrated through that route while they were migrating. Some natural calamity happened and everybody perished in the lake. When we analyzed, of course, the samples were 800 to 1200 year old. In fact, we found there were three clusters, and one of the cluster was saying that they are the local people who are in the ground level or sea level, the base of the Himalaya. Second one was surprising because that was the Mediterranean people who probably I don't know why they have gone there. Third, at least only one sample which was clustering with Southeast Asia. There's an opportunity. We should go there, collect almost all the samples and analyze them and come to a major conclusion. So that's what we are thinking in the future.
who are these people? So most accepted speculation was that they are the victim of epidemic or they were traders coming from Tibet. But when we got genetic data, I got surprised because I got two clear-cut ancestry in that Rupun lake. One ancestry was matching with the present-day population of Greece, and second ancestry was matching with the population, present-day population of North India. We did the dating of all the individuals, and we found that there was a two catastrophic phenomena that has happened in the Rupun Lake. One happened in 7 to 8th century AD, and the second phenomena happened in 16 to 17th century AD.
The results contradicted previous assumptions that all these skeletons were deposited at the same time. Instead, the two major genetic groups were actually deposited approximately 1,000 years apart. But we are forgetting one thing here that's very important. Collaborations lie at the heart of such complex research. It's not just about scientists working in isolation. To draw significant conclusions, anthropologists, archaeologists, linguists, and historians all play vital roles. In one such collaboration, geneticists including Thangaraj and anthropologists from Punjab University used DNA and isotope analysis in combination with historical records to shed light on skeletal remains excavated from Punjab's Ajnala in 2014. DNA sequences matched with people from Uttar Pradesh, Bihar, and West Bengal states in India, while the historical context suggests that the remains might have belonged to Indian sepoys killed by the British during the First Indian War of Independence in 1857.
So all these gives us a clue to connect with the modern human or contemporary human. So that's the starting point. Then that gives us more opportunity to explore the biological material using our genetic expertise to explore more and then connect that genetic information with additional genetic information either from ancient DNA or from the contemporary DNA and connect.
Neeraj also traveled extensively, meeting archaeologists and historians and showing them the emerging potential and importance of this field.
And over last 5 6 years, I made like more than 100 collaboration Southeast Asia and some region of Middle East also. More discussion with archaeologists, the idea of prehistoric humans, how they have adopted the agricultures, and what was the subsistence pattern, and what was the relation of ancient humans with the pastorals, how they have migrated together, and how they have cope up with the climatic change, how they've adopted different type of agricultures.
Ancient DNA science has this cool factor that really grabs people's attention. It's a big reason why there's so much public interest and why partnerships keep popping up. Everyone wants to be part of uncovering these ancient mysteries. Ben Lamb, the CEO of Colossal Biosciences, actually gets fan mail and DMs, mostly from kids and their parents. Imagine that.
I'm not from the conservation space. If you ask me to go raise capital to go save a specific species, I don't know if I could do that. But I think building a team to go after this like moon landing type thing and rally behind it, where kids and parents are excited. We get emails and DMs on all these different platforms, and even physical mail where like parents have sent us things that their kids have drawn, and they're like, "Thank you for doing this."
People are going to be curious. People are going to want to know why you're doing this. What's the point? They had never thought that their trophies could be used to extract genetic material, right? But so I was the first person who was kind of coming and telling them this stuff, right? But once they've heard it and I've interacted with them, they do slowly open up. And then the great thing is the amplification which happens, because they know others. I mean, India is like a large community, right?
Uma here is taking her ancient DNA work to museums next. She teams up with wildlife historian Raza Kazmi to interview descendants of the former royal families. These oral histories reveal much about their hunting practices and the wildlife that once thrived in these landscapes. Here is politician T.S. Singhdeo, the titular Maharaja of Surguja in Chhattisgarh, in one such chat with Reza.
We were told that there would be tigers with larger heads, wider chests, smaller tails. This was a characteristic, a distinct characteristic of tigers in the northern belt of Sarguja. Then there were the in between variety. And then you would have the leaner variety, let us say Manpart and some other areas, where they would be slender, sleek, long, with reasonable size heads, balanced body structure, shoulders. Not that the head and body is much larger compared to the rest of the body and the tail.
Meanwhile, Thangaraz is also excited at the prospect of precision medicine by understanding how past genetic adaptations influence present day health. From pinpointing disease risks to tailoring medical treatments, this research is shaping the future of personalized healthcare, all while connecting us to the distant past. It's like decoding history for better health.
So when the community strictly following the endogamy marriage or marrying within the group, if there is a mutation in either in the father or the mother, it's going to go to the next generation. If the mutation, the recessive mutation is in one of the chromosome, that individual absolutely fine. He or she is transmitting to the next generation, and that individual also will be normal. So it happens generation after generation. That particular mutation, if it exists in one of the chromosomes, it's going to well spread in that particular population. So after few generation, if the mutation exists in the heterozygous condition, that means one of the pairs going to spread across the population. Then after few generation, the father and mother who carry mutation in one of the chromosome, there's a possibility that they contribute the abnormal chromosome to the next generation. Therefore, the next generation has mutation on both the chromosomes. Hence, there is a disease. That kind of situation we predicted is going to be very high among Indians. In fact, we did find such kind of mutation exist in that particular population and causes disease which are severe, and it leads to very, very early death, less than 2 months of after the birth. Children are dying. That kind of situation is quite high among Indian populations. So there was no record so far, but genetically we predicted that one third of the populations are having such kind of mutation. In fact, we identified that in some other population, and which we are now trying to screen the entire population to evaluate who are the people carrying the mutation, and we are thinking of giving counseling. Therefore, this disease can be avoided in the next generation.
Well, ancient DNA coming to the rescue here. That's a wrap on today's dive into ancient DNA research. We've explored how this cutting edge science is uncovering our past, reshaping our understanding of human history and conservation, and even influencing modern medicine. Our story began with death, but it ends in hope, thanks to the efforts of these brilliant scientists. Stay tuned for the next episode in this series of Biolore. Till then, you can go back to all the past episodes of the Nature India podcast on your favorite podcast channels.
On the sands of time, stories unfold, treasures from the past to modern lore. Every leaf, every cell, life secrets they tell. Biolore.
This episode was brought to you with support from the National Centre for Biological Sciences.